Just let me know. Can you see the screen, children?
Yes, ma'am.
Yes, ma'am.
And my voice is also audible now.
Yeah.
Yes, ma'am.
Okay, thank you. Let us begin with this. Last time we have discussed first and second law, right? So monohybrid cross also we have learned. Monohybrid ratio also we have studied. Mendel's first law, that is law of dominance, we have discussed. Second law, that is law of segregation, or also it is called as law of purity of gametes, that also we have discussed. Now we'll come to the third law, that is law of independent assortment, which we have already discussed. I have shown you this example also, which can be explained only with the help of dihybrid cross. See, sometimes the question can be asked indirectly. Explain the law which is dependent on dihybrid cross or dihybrid ratio. Then in that case, you should understand that this is the third law. The question will not be asked directly as explain third law or explain the law of independent assortment, but it can be asked as explain the law based on dihybrid ratio or dihybrid cross. Then in that case you should understand that is the third law, and that is law of independent assortment. I explained you that day in short, but I want to explain it, discuss it again in detail. Di means two hybrids in a cross when the cross is between the parents differing in two characters, when two characters are considered for the hybridization method. So that's why this is called as dihybrid cross, right? So when parents having considering two different characters in a cross, that cross is called as dihybrid cross, and the ratio which we get with the help of this cross is called as dihybrid ratio. Now what are these two characters considered here in this example? One is color of the seed, that is yellow and green. Yellow is dominant, considered to be dominant, and green is considered to be recessive. That we will see, and accordingly the genotypes also are mentioned in this example. And the second is what? The second thing is what is considered in this case is the texture of the seed, whether it is smooth, round shapes. It is considered to be shape in the textbook as per the text shape, that is whether it is round or wrinkled, having wrinkles on the surface of the seed. So
Ma'am,
accordingly, yes.
can you please admit Srishti Joshi?
What are you saying? I didn't get.
Can you please admit Srishti Joshi? She
Now
messaged
wait.
me.
Yes, yes. Wait, ask her to wait. I have to admit it through another device. I will start my another
Yes.
device. So just ask
Ma'am,
her.
she said there is some network issue, so she can't.
Yeah, yeah. I'm starting my device. Just ask her to wait for a few minutes. Okay.
Okay, ma'am.
Yeah.
Thank you so much, ma'am.
Yeah, welcome. Actually, I was going to, but I forgot. Why when I started talking with you, I forgot to start my device. Yes, admitted. Now my another device is also on. I will admit the latecomers. Please join on time so that this will not happen. Okay. So what we were talking about the dihybrid ratio and the dihybrid cross. Here, two characters are considered. Two contrasting characters are considered. Now we will discuss this law again. Okay. What is this now? Two characters are color of the seed and the shape of the seed. Okay. Color of the seed, yellow and green. Yellow is considered to be the dominant and green is considered to be the recessive one. Whereas round shape of the seed is considered to be the dominant over wrinkle type. So these are the phenotypes, and genotypes, how they are represented as YY and capital R for the yellow and round. Whereas for green and wrinkled, small y, capital small r. So these are the recessive. Okay. Now what are the gametes? Capital Y and small r. Y for yellow, R for round. Same here. Small y for green and small r for wrinkled. Then these gametes in the F1 generation, whatever the offsprings are produced by crossing these two parental forms, all the hybrids or all the F1 offsprings, they are seen to be with yellow colored and round shaped seeds. So all the offsprings are showing the seed color as yellow and shape as round in F1 generation. Now when this F1 generation is allowed to self pollinate, then what happens? It is producing how many types of gametes? Four types of gametes. Now you can see these four types of gametes. First is capital Y capital R, second is capital Y small r, third is small y capital R, the other fourth one is small y small r. So you can understand here independent assortment here itself, because first capital Y small capital R and the last one small y small r, these are only having these gametes present in the parent, original parent, but these of the connectivity also. Are you able to hear me,
Yes.
children? Just
Yes,
let
ma'am.
me. Ha,
Yes.
so I have connected another one. Yeah. Okay.
Yes,
So what I
ma'am.
was telling here itself, you can see now when the gametes are formed, when this F1 hybrid or F1 plants offsprings are allowed to self-pollinate, you can see that these two are totally different. These two gametes are having the genotype different than their original parents, and that is what called as independent assortment. Means independently they are assorted when two characters or more than two characters are considered at a time in the parents, right? When only one single character is there, then they show their genotypes to that of their parents, either of the parents. But here you can see when two characters are considered as yellow, green and round wrinkled, then two more types of gametes are formed which are totally different than their original parents. This is what called as independent assortment. Now when we observe it with the help of Punnett's method, Punnett square method, or this is also called as checkerboard method, how it is shown, these are the male gametes and these are the female gametes in F2. When F1 is allowed to self-pollinate, now you can see same gametes here are shown horizontally as male gametes and vertically as female gametes. Now, what is the combination of one male with the one female? Now, see, this will consider the one female from left hand side, the first one capital Y, capital R, and also the first in the transversely arranged male gametes, that is capital Y, capital R. Here, what is the result? You can see all capital, that means all are dominant. Then, considering the same female gamete with the gametic genotype, that is YR capital both, and the second male gamete as capital Y small r. This is the result. Again, this first one with the third type of gamete with the genotype small y capital R, then the resultant progeny is capital Y small y and both capital R. Then the fourth one, when it is crossed, the result is y small y capital R small r. So in this way, then again the second female gamete with the first male gamete, second, third, fourth. In this way, when you see cross each one, then this is the resultant, and it is shown with the help of different colors also purposely. So how it is seen now, we will discuss the with the help of these gametes, we will count the number of those type of seeds. Now, when both are capital or at least one from each one is capital, that means that particular character is dominant over the other. So the first one is yellow round, second one also yellow round, because both are capital here Y and one R is capital, so this is also yellow round. The third is also yellow round. This is also yellow and round. Then this is the fifth one. At least one is capital, one letter is capital out of these two. So this is also yellow and round. Then this one is also yellow and round. So how many? Six. Then this is the seventh one, yellow and round. This is the eighth one, yellow and round. This is the ninth one, yellow and round. So yellow and round, how many? 9 seeds, means now 9 offsprings out of 16 they are showing yellow and round seeds, color as yellow and shape as round. Now the second category is here, you can see with the help of genotypes. Y is both capital, but here are both are in recessive form, means when both are in recessive form, that means what should be the phenotype? Wrinkled, obviously, you know, as per the previous original parent, it is wrinkled, and yellow color is Y both capital. So what is the phenotype of this seed? This is yellow and wrinkled. Then how many yellow and wrinkled? Here also yellow and wrinkled, 2. And then the third one as yellow and wrinkled. So yellow and wrinkled, which is not the related to the original parents. Yellow and wrinkled is totally new form, or you can say phenotype, which is produced. Now the next is what? Third one, when both Y are small, but R, either both or one R is capital. That means what? This is green and wrinkled. When both the Y are small, as per the original parent, you can see that the color is what? Green. So green but round. One of the R is capital. That means it should be, whether it is homozygous or heterozygous form, it is showing which shape? Round shape. So this is yellow round, sorry, green round. This is also green round, and this is also third also green and round. So total 3 green and round out of 16 they are produced. And the last one, that is original parent, recessive parent, that is green and wrinkled, only one. So phenotypically when we see, not only the two like original parents as yellow round and green wrinkle, but two more phenotypical expressions are seen in the F2 generation, that is yellow wrinkle and green round, which are assorted independently. What these are totally different than their original parents. So that's why this is called as law of independent assortment. So when dihybrid cross is performed, in that the phenotypic ratio of dihybrid cross is generally always 9:3:3:1 in Mendel's experiment. Whereas genotypic ratio, we will not concentrate much more on this, because you can see, you can count it, pure totally homozygous dominant only one, homozygous recessive only one. You can see the first and the last one, and then accordingly you can count the remaining. Okay, so that is different. So we will not count right now genotypes, but what we are going to concentrate is only on the phenotypic ratio. So when dihybrid cross is considered, the phenotypic ratio is always 9:3:3:1 in Mendel's experiment. So remember that. Okay, now we'll come to the next part of this chapter, that is called as back cross and test cross. Now what is the meaning of back cross and test cross? I'll tell you. Now you have, in the previous experiment you have observed whether it is monohybrid cross or dihybrid cross. We will, for our convenience, will take the example of monohybrid cross only. So in monohybrid cross, you remember that mono means single, only single character is considered. Then if you remember that height is considered as the example, a character for crossing. Then all the F1 progeny were tall, but the genotypic ratio was capital T, small t, that means heterozygous form, but phenotypically they were all tall. That means this F1 progeny, when is crossed back again with either of the parents, then that is called as back cross. Try to understand. F1 progeny, when is crossed back with either of the parents. Either means one parent is with both capital capital T, and the other parent is pure of homozygous recessive. This is homozygous dominant and this is the homozygous recessive. That means F1 plant which are heterozygous with capital small t, when are crossed back, any of the parents homozygous dominant or homozygous recessive, that means pure tall or pure dwarf, then that cross is called as back cross. Whereas test cross is when F1 progeny, means F1 offsprings, when are crossed back with homozygous recessive parents. That means only pure dwarf parents. That means capital T small t, when it is crossed with small t, then that is called as test cross. So that is the difference between the back cross and test cross. Test cross can be back cross. Every back cross cannot be test cross, because test cross is the cross when F1 is crossed, one offsprings or progenies are crossed back with only homozygous recessive parents. What is the importance of this test cross? It is very important to use the test the nature of the hybrid. That means when we cross the F1 progeny with the homozygous recessive, then we come to know the genotypes of the parents also. So, and it is a type cross only, and it is useful to introduce useful recessive traits in hybrids of self-pollinated plants during rapid cross improvement programs. And the recessive parent is crossed to find out the unknown phenotype. Now, unknown is what is not, we don't know the phenotype of that particular parent. So when we cross back to the homozygous recessive, how it looks, that phenotypic expression we can come to know. And that can be explained with the help of the same example of pea flower, where you can use the homozygous recessive parent with the homozygous recessive parent. Now that I will explain this with the help of another example of test cross in the another slides. But before that, right now I don't have this slide immediately with me. I will show you in the another slide. Now the next part is called, considered to be new Mendelism. That means Mendel first of all performed all his experiments. He proved some important laws, first law, second law, third law, he explained. But along with that, considering Mendel's experiment, few more scientists, they perform their experiment using different what you can say experimental what you can say experimental, maybe plants or animals. Okay, so the other scientists also perform their experiments considering the results and the laws proved or explained by Mendel, and that is considered to be neo-Mendelism, new than something whatever Mendel proved at that time using his pea plants. Okay, then what are these things? That all these things come under post-Mendelian era called as neo-Mendelism. So first of all, Morgan explained using the experimental device or experimental animal or insect, you can consider as the Drosophila melanogaster. Also, he used the plants like Mirabilis, Jalapa, evening primrose, etc. Some scientists, they perform, group of scientists separately or together, they performed some experiment which were similar to the Mendel, but using Mendel's experiments, they could prove some few more uh, uh, terminologies or few more things like the expression of the plants which are not exactly similar what Mendel proved with his experiment on pea plants. And these are some of the examples which we are going to study. So what are these modified things explained by other experiments by different scientists is, these are considered to be gene interactions. There are two types of interactions given in your textbook, that is intragenic and intergenic. Intra means within the species or the alleles, and intergenic means in between the different. So intragenic expressions, they occur between the alleles of the same gene. For example, incomplete dominance, codominance, multiple allele series of a gene. These are the intragenics, means within the gene, same gene, but the alleles are different. So that is what called as intragenic. And under that, we will be studying incomplete dominance, codominance, and multiple alleles also. Now, first, one by one, we'll discuss about the incomplete dominance and codominance. It is very easy to remember also and understand also. Incomplete, which is not complete. That means, if you remember the first law of Mendel, what is the first law of Mendel? In F1, when that is called as law of dominance, right? Law of dominance, what it proves? When two parents considering a single character are crossed with each other, then F1 generation, only the characters which are dominant are expressed, and the characters which are not expressed, they are called as recessive characters, which are suppressed. So when height is considered as per Mendel's law, tall and dwarf, tall and pure dwarf plants, when are crossed in F1 progeny, all plants were tall, but the genotypic expression is heterozygous. That means one is capital T, the other is small t. But rather than this, some deviations from the Mendel's experiment and Mendel's laws are also observed and proved by different scientists using different plants or animals. Okay, so this is what one of the examples of from Mendel's deviation is. Whatever the laws were proved or explained by Mendel, these are deviated from the original, what he proved, what Mendel proved. So this is one of the examples, that is incomplete dominance, which do not prove, which cannot prove the first law, that is law of dominant. As per the first law of Mendel, law of dominant, or when we are considering this example, all the F1 progeny are showing the dominant characters. But here, when this is explained with the help of incomplete dominance, this law cannot be proved. It is here the flower like primrose, or the example of evening primrose, this is also called as, such plants when are considered, then in this case, these flowers, they are of two types. One is red colored flower and the other is white colored flower. Okay, in this example of incomplete dominance, where Mirabilis jalapa is considered the evening prime plant, where two contrasting characters are considered, where flower with red color and the flower with white color. So phenotypically, one is red, the other is white. When these two are crossed, these plants having one with red colored flower and the other with the white colored flower, they are crossed, the resultant ratio, that means the resultant progeny and in F1 was expected to be all red as per Mendel's law, but it is not observed. But what is observed? All the flowers in the F1 plant progeny were pink colored. That means what? Neither red nor white, but it is the combination of these two characters, that means pink. So F1 progeny is not showing the complete dominance of either red or white, but incompletely red and white are expressed in F1 generation, which gives the pink color, and that is what called as incomplete dominance. Neither red nor white is expressed in F1 generation. Incompletely it is expressed, partially it is expressed, so that's why it is giving as pink color. So that is what called as incomplete dominance. When these F1 plants are allowed to self-pollinate, then you can see here, these are the male gametes and these are the female gametes. Two types of gametes are shown because this pink color is having one capital R and one small r. So two types of gametes are produced. When these plants are allowed to self-pollinate, these two gametes, what they are showing results in F2 generation, red color, one flower, capital R, second capital R, small r, third capital R, small r, which is expressing, giving the phenotypic expression as pink colored flowers, and the last one, small r, it is giving white flower. So what is the phenotypic ratio in case of incomplete dominance? That is, red is to pink is to white as 1:2:1, and this is the example of first example you can consider as the deviation from Mendel's first law, which is deviated, which is going away from the original law, which the first law of Mendel cannot be proved. First law of Mendel cannot prove this example. So this is one of the examples of Mendelian deviation, or this is the best example of incomplete dominance. It is neither of the parent is dominant in the F1 generation completely, but incompletely it is showing, that means partially it is showing its expression. So this is the example of incomplete dominance. Now, the second example from Mendel's deviation is codominance, which are the examples of intragenic inheritance also, right? Within the gene, but the alleles are different. Now, another example here is the codominance as cattle coat color. Cattles you have observed, cattles, any cattle you can consider, maybe a cow or any other. Now when we are considering any of the cattles with red colored coat, and other one as white colored coat. Okay, one is red coat color, having the red coat color, and the other parent is with the white coat color. Now here, what are the genotypes shown here? You try to understand. For red, both capital letter are used, and for white, unlike this red and white flower, here both W genotypes as both W capital letters are used. It is not small r. Try to understand, this is the difference in this example as codominance. Okay, so cattle with red color coat is represented as capital R, whereas with white colored coat as capital W for white, capital W. Then these two parents are crossed in F1 generation. You can't see the combination of these two red and white color, but what you can see, that some patches are of red and some patches are of white. So capital R and capital W means, what is the difference, or how it is different than the first previous example? That here both are expressed. Red is also expressed and white is also expressed in the pure form, right? So totally red or totally white is not there as per the Mendel's first law of dominance, as well as it is not like the mirabilis jalapa flowers as in incomplete dominance. That is not the combination of red and white color. Pink color drone is not seen, or any other color of combination of red and white is not seen in the cattle coat, but both the colors are expressed. So this is what called as roan color, means red is also expressed and white is also expressed. So these cattles are considered to be roan colored cattles, means both are expressing, both the parental characters are expressed equally in this case, and so this is called as codominance. So cattle coat color is the best example of codominance, whereas mirabilis jalapa, pink colored flowers, is the best example of incomplete dominance. So flower or the plant is the example of incomplete dominance, and the animal as red and white colored, that means roan colored cattles, is the best example of codominance. Codally, both are equally expressed, so giving the color of coat of the cattles as roan means both are expressed, red also, white also. Now when these cattles are allowed to self-pollinate in F2 generation, again these many combinations as red, roan and white, means genotypic as well as phenotypic ratio is 1:2:1 in codominance, as well as In incomplete dominance also, phenotypic and genotypic ratio is same as 1:2:1. So this is also one of the best examples of Mendelian deviation, which do not prove Mendel's first law. Okay, so this is the best example you can understand. Now the next example of intragenic inheritance is multiple alleles. Multiple means not only two but more than two alternative forms of alleles of a gene in a population occupying the same locus on a chromosome or its homologue. That is called as multiple allele. That means here you can observe only two types of color here considered. But here if you see multiple, many alleles are present for the same gene. The gene which is expressing the characters is showing the more than two alternative forms. Now the best example of this multiple alleles is the Drosophila melanogaster, that is the fruit fly, common fruit fly, which you know its name is Drosophila melanogaster. And another example of this is ABO blood group system in humans. Now we will see what happens here. Because of these many forms, alternative forms of the alleles, there are chances of mutations. Wild type of gene is present already there, and that wild type of gene undergoes a series of mutations. That gene can mutate several times, and that produces so many alternative forms or expressions, and in a series that shows the dominant recessive and along with that codominance or incomplete dominance among themselves also. So wild type is dominant over all the other mutant alleles. Now the best example, as I told you, that Drosophila and ABO blood group system in humans, we will discuss what is that exactly. Now Drosophila melanogaster, if you see, this is the first uh, fruit fly with the normal wings. The size of the wings is normal, shape and size is normal. So that genotype is expressed by VG plus. Okay. Whereas when this gene gets mutated, then there is change in the shape and the structure shape of the wings, which becomes that nicked wings. They are called as nicked wings. Shorter. They become shorter. VG. They are represented by Ni. Okay, normal wild type by plus and then nicked one by Ni. Then notched, again it that mutated form, it shows the wings of notch type, that is represented as genotype No. Then strap wings, it is represented by ST, and vestigial wings mean without any wings, they are reduced one, without wings or no wings or no a very much reduced form, or which is as good as no wings here, that is VG without anything. So these are all the forms, different forms. These are all mutated forms, but the normal wings, the Drosophila melanogaster with the normal size and shape of the wings is considered to be the wild one, whereas by mutations it produces so many forms. These are all alternative forms. So this is what called as multiple alleles, the example of multiple alleles. Many alleles are seen for a single gene representing the same character, that is the shape of or the size of the wings, right? So this is what the example of multiple alleles. Now the second example, which you know everybody, that is ABO blood system in human. You know, ABO blood system means what? In human beings there are basically four types of blood groups, that is A, B, AB and O. But for these four type of blood groups, there are not only four types of genotypes, but more than that. That means six type of genotypes are observed, right? So like Drosophila here, only one normal wing is a wild one, but remaining all are mutated ones. Like that, in ABO blood system, phenotypically only four blood groups are there, but genotypically there are six. Now, what are these and how it is? The allele, that means gene I, represents that immunoglobulin. okay, controls the ABO blood groups. It has three alleles, that is IA, IB, and small I, or this is also called as I0. Okay, so the allele A and B produce slightly different form of an antigen, and allele small I does not produce any type of antigen. Since humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are codominants and are completely dominant over I. They are codominant. And you will be telling me which is the example of codominance in this example of blood group. There are 6 different genotypes and only 4 different phenotypes, that is blood groups. Now see, when the blood group is A, what are the genotypes? IA or IA small I. As I told you that humans being diploid, they show in this way, pair form only. So either homozygous dominant or heterozygous nature for A, which that immunoglobulin, which is a protein, produces A type of antigen. So represented by IA or IA small I. Then B group when is present, what are the genotypes? IB or IB small I. This is the heterozygous condition. This is the homozygous condition. When the blood group is AB, it shows the presence of both the antigens IA, IB. Now you can tell me which is the example of codominance. And when there are no antigens produced, then this is the homozygous recessive form, that is blood group A without any antigen, both in recessive form, homozygous recessive condition. So phenotypically only 4 blood groups are present, but genotypically there are 6 genotypes present. Okay, so this is the best example of multiple alleles, which is considered to be the deviation, one of the deviations from Mendel's laws. Now we'll talk about the next part, that is the linkage and crossing over. There are several genes present on the chromosomes. Chromosomes are considered to be the carriers of heredity. Genes have tendency to inherit together, called linked genes, and this is called as linkage. All the genes linked to one another in a particular chromosome constitute a linkage group, and number of linkage groups of a particular species corresponds to its haploid number of the chromosomes. For example, Drosophila, it has 4 pairs. Garden pea, 7 pairs of contrasting characters, as we have learned. And in human beings, total, how many pairs of chromosomes are present? 23 pairs of chromosomes are present. That is the haploid condition, which is shown here, the number of chromosomes in haploid form. You know, haploid means single set. Diploid means double set. Triploid means 3, like that. Now we will see what exactly this kind of linkage is. There are 2 types of linkages. One is complete linkage and the other is incomplete linkage. Now what does it mean? Complete linkage means genes which are closely associated in the chromosome, they cannot be separated because they cannot be crossed with each other, because they are very closely associated. The genes on the chromosome, they are very much closely associated with each other, present very close to each other, so cannot be separated and cannot be crossed, but inherited together. So this type of linkage is called as complete linkage, because they are very much close to each other. As it is, they are completely inherited, they cannot be crossed, there are no chances of crossing over and separation. So these are considered to be the complete, that type of linkage is called as complete linkage. An X chromosome of Drosophila males, that is the example. Then incomplete linkage means what? The linked genes are distantly located on the same chromosome and have chances of separation by crossing over. And so new traits occur in the offsprings. That means on a chromosome, when genes are loosely arranged, they have they are arranged distantly, they are located distantly on a particular distance. When they are located, then only there are chances of crossing over, and then only there is a possibility of separation and crossing over, and that's why the new traits or new combinations can be formed in the offsprings, and that is what called as incomplete linkage. They cannot be inherited as they are, but they, as these genes are distantly arranged located on the same chromosome, there is possibility, there are chances of crossing over and separation, and there is that's why new forms, new traits can be observed in the offsprings. So example of this incomplete linkage is Z maize color and shape of the grain. When you observe the Z maize is what you observe maize. If you observe the grains of the maize, you can observe the different colors in the maize as well as shape in the grains. So that is the example of incomplete linkage. Now the next point is sex linkage. Sex linkage means what? The definition is as it is, I'll tell you right now. Passing of or transmission of body characters from parents to offspring along with sex is called sex linkage and is called sex linked inheritance. Very simple to understand. Whatever the characters are present in the parents, when they are passed on to their offsprings along with the sex, that is sex linkage and is called as sex linked inheritance. Means body characters as well as along with the sex of the child, when the characters are transmitted from parents to the offsprings, then that is called as sex linkage. Now there are again two types, like normal sex linkage, what we were talking, the same here you have to remember: complete sex linkage and incomplete sex linkage. Now here also similar things are to be considered, taken into consideration. That means complete sex linkage is what? When genes located on non-homologous region of X and Y chromosome inherit together because crossing over does not occur in this region. Now try to understand. Now I will explain, when I'll explain the structure of chromosome, it will be very easy for you to understand. See here. This is the structure of chromosome. This is the X chromosome and this is Y chromosome. Okay. Now this is the centromere, point of attachment of these two chromatids. A single chromosome is made up of two chromatids, this one and this one, parallelly growing one. And this the point of attachment, this is called a centromere. Okay, this longer one is called as X chromosome and this shorter one is called as Y chromosome. And you can see from the centromere here itself, this part is similar in both X and Y chromosome. So this region is called as homologous region. Homo means similar, logos, that is morphologically similar region present, that is in X as well as Y chromosome is called as homologous region. Whereas this part above the centromere, this is long and elongated in case of X chromosome and this is short here in case of Y chromosome, which is not similar morphologically. So this region is called as non-homologous region or non-homologous part, which is dissimilar in both X and Y. This is different than Y and Y, this part is different than X. So that's why this is morphologically dissimilar, which is not similar. So this region is called as non-homologous region. Now what we were talking is about the incomplete and complete sex linkage. Now what is complete sex linkage? When genes located on non-homologous region of X and Y chromosome inherit together because crossing over does not occur in this region. Here in this diagram, if you see here, the genes cannot be crossed with each other because of dissimilar morphology. Morphologically they are not similar, so that's why no chances, no possibility of crossing over in this region. And that's why these genes, they are inherited as they are in the next generation. So X will come in X and Y in Y, like that. They will be inherited as they are. There is no chance, there are no possibilities of crossing over as dissimilar nature, as of its dissimilar nature. That's why there is no crossing over, and that's why this is called as complete sex linkage. So example of this is hemophilia, red green color blindness, myopia, hypertrichosis, ichthyosis. Now see, hypertrichosis is a very common example you can remember for the complete sex linkage. That means Y, this which is related to Y gene only, and this is these two examples which are related to X gene only. That means X chromosome only. Means hemophilia, red green color blindness, myopia. These are the examples related to X chromosomes. They are present only on the non-homologous region part of the X chromosome, which are inherited as they are. Okay. Whereas hypertrichosis and ichthyosis, these are the genes located on the non-homologous region of the Y chromosome. So they are located only on non-homologous part of the Y chromosome. So that's why they are completely inherited as they are from Y. Okay, so that is this is the best example. In the further examples, I will explain what does it all mean. So just try to understand what complete sex linkage means. The genes which are present or located on the non-homologous region of X and Y chromosome, they inherit together because crossing over does not occur in this region. And these are the examples of X chromosome, that is the non-homologous region of X chromosome, and Y as non-homologous region of Y chromosome. Now, what is incomplete sex linkage, that we will see. Here, this happens by genes located on homologous region of X and Y chromosome, which do not inherit together because crossing over occurs in this region. And these are examples are XY linked traits. Means XY linked traits, they are called as XY linked traits. And the examples are total color blindness, nephritis, retinitis pigmentosa. These are the examples. What exactly that is, that I will explain in the later stage. Just try to understand now this example, the diagram again. Now here is the region called as homologous region. Now for incomplete dominance, which we are talking, here is the homologous region where there are chances of crossing over in this region. Morphologically, this region is similar. So in this region, there are chances of crossing over. There is possibility of formation of new traits. And because of this crossing over, this is considered to be incomplete sex linkage. That means completely they are not inherited as they are, but they do not inherit together. They do not inherit together because of crossing over. There are chances of crossing over. So these genes present on this homologous region do not inherit together. And these are some of the examples. Now we'll talk about crossing over. Now what exactly crossing over, that I will explain in the next period, which happens during the meiosis. For that, you need to learn the meiosis part, but I will not explain what exactly that meiosis part is. Just try to understand this part first right now, that is X chromosome and Y chromosome. X chromosome is straight, rod-like, longer than Y, metacentric, has more euchromatic. Now I have already showed you this diagram of X and Y chromosome. It is longer than Y. This is shorter, this is longer. So this region is larger. So more number of genes are present in X chromosome than Y chromosome. Then even one more thing you have to remember, that it has the euchromatic region more in X chromosome and less in Y chromosome. As the region is larger in X chromosome, the euchromatic region means what? More DNA is present in X chromosome and Heterochromatic region is less, which is less active genetically. Euchromatic region or euchromatin means the active form, genetically more active, which is present more in X and less in Y. Whereas when we consider this as the metacentric, means the centromere when is exactly present in between, at the center of the chromosome, making the two arms in the equal form, that is called as metacentric nature. Whereas in case of Y chromosome, it is not metacentric, but unequal arms are present here. That is called as acrocentric form of uh, uh, Y chromosome, where one is shorter arm and the other is larger arm. And here in Y chromosome, heterochromatic region or heterochromatin is more in Y chromosome, that is non-genetic region is more, whereas euchromatin region, which is more active, where DNA is more, that is less. So genetically Y chromosome is less active than the X chromosome. So this is what about the structure of X and Y chromosome, as well as linkage, incomplete and complete linkage that we spoke. Here we will stop.